My name is Denise Sheer. I'm Professor of Human Genetics and I work at this amazing institute, the Blizzard Institute, which is part of Barts and the London School of Medicine and Dentistry, and we're part of Queen Mary University of London. My interest in brain tumours started about 20 years ago when I was doing research to try to understand the genetic basis of children's cancer in general. And neuropathologists started sending me tissue samples from brain tumours to try to understand the genetic changes that are in those tumours. And unfortunately the technology wasn't really good enough for us to find anything. But over the years the technology has really improved greatly, and now we're able to dissect the genetics of these cancer cells in much greater detail. The focus of our lab's research is to try to understand what are the molecules and the pathways in the cell that makes a normal cell become a brain tumor cell. And we do this at different levels. We do this at the level of the DNA. DNA codes for RNA in the cell, and RNA codes for protein. So we can look at all these different levels ultimately to find out how the proteins, the abnormal proteins, then will give rise to a cancer cell. So we're working on pediatric low-grade gliomas, and these are relatively common tumors which are found in children. And what we're trying to do is to examine this pathway of DNA coding for RNA coding for protein. And this is on the basis that if you've got abnormalities in the DNA or in the RNA or anywhere along the process, you're going to get abnormal proteins made, and those abnormal proteins are going to give rise to the cancer. So what we're trying to do in this particular instance, in the past what we've done is we've done a lot of studies on the DNA of the brain tumour cells, and funded by the Brain Tumour Charity actually, we made key discoveries in the genetic changes of low-grade gliomas. Now what we're doing is looking further along the pathway, because we know that there are RNAs called microRNAs that are crucial also in regulating the types of proteins that are made. So here for this one, this project, what we're doing is we are looking specifically at the microRNAs in a group of tumours where we already know the genetics, and we're going to be putting all the data together to derive a really thorough understanding of how these individual tumours form. Proteins are important because proteins regulate every single process in the cell, and that from forming the structures of the cell to the molecular pathways that allow a cell to change in response to its environment, and also for a cell to just normally function. What the grant from the Brain Tumour Charity will help us to achieve is to derive a really thorough understanding at all these different levels of the cancer process that leads to low-grade gliomas, and to the different kinds of low-grade gliomas. So for example, you might have one kind of low-grade glioma, like a pilocytic astrocytoma, that will have a particular genetic change at the DNA level, and then will have different changes along the pathway to a protein. You might have another tumour that's a diffuse astrocytoma that will have a different genetic change, and in turn also a different change in the RNA. So this will allow us to derive a thorough understanding of the different types of pediatric low-grade gliomas. I hope that the greatest benefit will really be to try to understand that the scientists and the doctors can understand what are the molecular pathways that lead to the formation of the tumour. Secondly, I hope that we will be able to make discoveries that will allow new forms of treatment to be derived from what's called the targets that we're identifying in these cells. Thirdly, I'm hoping that understanding the molecular basis of these different types of low-grade gliomas will allow a new kind of subclassification to be derived. The major challenge facing the brain tumour community in the first instance is sufficient funding. We really need more funding to be able to fund more projects. Thinking about the projects that are formed, we really need to be able to understand the scientific basis, the molecular basis of how these tumours form. We then need to be able to have really good interactions with the clinicians, and so that in time we can develop treatments which are based on these molecular findings and which would help the clinicians in providing new forms of treatment for patients. Secondly, thinking about the process of a normal cell forming a cancer cell, There are many different steps along that way, and we now know that there are many pathways which are affected. We need to be able to develop techniques for understanding that complexity. I think the biggest breakthroughs will come from a subclassification of tumours. Once you've subclassified tumours in a molecular sense, then you can develop new forms of treatment based on the molecular targets that have been identified in these subgroups of individual brain tumours. The funding to the research community is absolutely essential. We need to be able to study brain tumours in all kinds of different ways. We need to understand what causes them to grow, to develop in the first instance, what allows them to respond to one kind of treatment or another. We also need to provide those essential links between basic scientists and clinicians. The funding that the Brain Tumour Charity can provide is also important in developing a really good community, a strong community with lay people, with scientists, with clinicians, all involved in the same purpose of improving the prospects for patients for brain tumours. The funding that the Brain Tumour Charity provides also gives an opportunity for young people to come into the brain tumour research community. For example, in my laboratory, for many years now, I've been bringing gap year students who straight from school, who have a year out, who come into my research program and start a period of intense they receive training, give them intense research opportunities, and they are all working on brain tumours. And after they've left us and go on to university, they maintain an interest, come back to the lab and keep participating in our research programs. Secondly, we bring medical students in. Every year we've got many medical students who come and work on the brain tumour program that we've got. I also bring master's students on to the program as well. And the aim is really to develop an interest, spark the interest in this really important area of research. I wholeheartedly encourage people to support the Brain Tumour Charity because the way they fund research is a great example of best practice. First of all, the projects are scrutinised thoroughly by independent peer reviewers. This leads to a period of interaction with the reviewers who suggest ways that the project might be improved. And this allows ultimately the projects that are funded to be the best possible that they could be. Secondly, the charity is supporting innovative research, and this is exactly what we need for brain tumour research. We need to bring in new ideas. Thirdly, what I think is really extraordinary about the charity and unusual is the way they bring the supporter groups into the charity. And people who are affected by a brain tumour and want to do something to help research into brain tumours can be rest assured that the Brain Tumour Charity are doing a really fabulous job in advancing brain tumour research. And one of the important things is that if the supporters are raising funds for brain tumour research, they know that at the end the research that is funded has been properly scrutinised.